next speaker from GlycanAge, Marina.
Hi.
Whenever
Hello,
you...
hi everyone. I'm sorry for the technical difficulties. The talk before me was also online, so let's head into it. So today I would like to talk about IgG glycans as biomarkers of health and aging. And before that, I need to tell you a little bit about the company that I represent. So today I'm talking in the name of Genus and GlycanAge. And Genus is the biggest high-throughput laboratory for the analysis of glycans in the world. So our clients are leading universities and research institutions, and out of over 191,000 published glycans, 159,000 were generated by Genus. So we're the highest contributor to the glycan data, and today I'm going to show you only a small fraction of this work. But before that, we need to talk about glycans, because this is largely a term which is not recognized even in scientific communities. So glycans are one of the four basic building blocks of life, including nucleic acids, proteins, lipids, and carbohydrates. So glycans are essentially carbohydrates. And when we imagine our cells, or when we see the cells even drawn in textbooks and university scripts and everything else, you often see the cell drawn with the nucleus, with the cytoplasm, and with a membrane, and that's where it stops. When in reality, on top of this membrane, you have a huge amount of proteins which are coated in glycans. So there is a huge fuzzy net of glycans on top of every cell. And glycans are the ultimate layer of molecular complexity. So they're post-translational modification, and they're most neglected molecules of cellular communication. So over the last couple of years, there's been a lot of talk about the spike protein. And the spike protein is often drawn as a protein, when in reality it is a glycoprotein. Just to illustrate what is the difference, I'm showing you here a picture of a naked bird without any feathers and a bird with feathers. So the bird without feathers cannot fly. But, feathers are not just there to make a bird pretty, they're actually there to perform some function. And glycans add a lot of functions. Sometimes, proteins cannot even do their job without glycans. Or just a simple switch of a glycan can completely change the function and the structure of this protein. What I'm going to talk about today is IgG. And IgG is also a glycoprotein. So IgG is the most prevalent antibody in our blood, and it's often drawn as a Y-shaped molecule, and here you can see its structure. And in this central cavity of the constant region, what you can see is that there is accumulation, basically the place where glycans are being connected, and where just by simply changing to different glycan structures, you can completely determine the structure and the function of this immunoglobulin G. The other important thing is that this is also a site on the immunoglobulin G where it's getting attached or communicating with other molecules and cells of the immune system. So meaning that, just a change of glycan on this site can determine the cascade of the communication within the immune system that is going to follow. So every person within their blood has a profile of over 30 glycan structures bound to their IgG molecules. And these different structures are present in different amounts, but they're between themselves, they are differentiated by the both number and the type of these small subunits, these building blocks that are assembled. So some of them will be, have more of these building blocks, and some of them will be shorter and have less of the building blocks. And this type and number of building blocks is what determines the properties of these glycans within the context of the IgG as a glycoprotein. So what we saw already in 2013, so this paper has been published in 2014 but it has been online since 2013. And what we saw was that glycans of the IgG actually make an exceptional marker of chronological and biological ages. So there were certain types, certain structures of glycans that would get increased amounts over the years, that would appear in increased amounts over the years. And the theory that we have behind it is something that is already generally known in the field, and it has been mentioned on this conference before, and this is the inflammaging. So basically all of the damage that is accumulated over the years and decades that we're living needs to be, it stimulates our immune system and generates slowly this chronic low-grade inflammation. And we can actually see this increase of a chronic low-grade inflammation in the change of the type of glycans which are present in people of certain age. So we can see that, for instance, young individuals have higher amounts here. The size of this glycan represents the relative amount. And we can see that actually younger people have higher amounts of these longer glycans that are extended with more subunits. And the older the person gets, the higher the ratio of these shorter glycans, which are pro-inflammatory. Well, what this basically means is that the glycans are showing us the chronic inflammation within one person's body. And we know that chronic inflammation is the culprit of modern diseases. It's basically present in all the, the chronic diseases, metabolic diseases, pulmonary, cardiovascular cancer, and so on and so on. They're all connected with the chronic inflammation. And when we look at the changes of IgG glycan, and this is true for many, many diseases. On this figure, I'm showing you some of the data, some of the cohorts that we've analyzed for systemic lupus, for ulcerative colitis, for Crohn's disease, rheumatoid arthritis, type 2 diabetes, colorectal cancer, and compare them with age, we can see that there is a general trend that is basically repeated both between these often age-related diseases and a simple aging. So individuals that have a higher chronological age will have more similar profile of glycans to the individuals that are younger but have chronic conditions. And this is something, we looked at this many diseases, but I don't have time to tell you all about all of those diseases. So instead, I'm going to show you what happens when we compare the glycan clock, so-called glycan clock, with other omics clocks and glycan clocks. And we saw, so this is a paper which was not published by us, it was by a different group, but what they saw was that glycan clock has a significant correlation with many conditions, including, cardiometabolic, pulmonary diseases, influenza, pneumonia, so even inflammatory diseases, and so on. And was best at predicting future hospitalization when compared to other clocks such as MetaboAge, clinomics, DNA, Horvath CPGs, Hanum, and so on and so on. And what we can see, for instance, when we look at one group of those diseases, for instance, the cardiovascular events, we can see that actually glycans are excellent at predicting cardiovascular events in a prospective cohort, and this was true both for men and for women. And in this paper, we saw that just a single glycan structure that is present on immunoglobulin G was able to predict the possibility of a cardiovascular event, as well as the AHA score, which is typically used. So just one single glycan structure was equally good at predicting cardiovascular events in women. And we cannot make a lot of the mechanistic studies in humans, but what we can do is we can look into model animals, and in this case into mice. And this was a collaborative effort with another group which is published in this circulation paper in 2019, where the mice, the obese mice, were fed a high-fat diet. And then what was done is basically that the IgG from these obese mice fed to a high-fat diet was put into the B minus minus mice. So those are mice that don't have their own IgG. And also in another group of those B minus minus mice, the control IgG was put. And simply putting this IgG from high-fat diet fat mice, these obese mice, which was hyposialylated, so it contained more of these pro-inflammatory glycans in comparison to the control IgG, they saw that basically the blood pressure was increased simply by adding a hyposialylated IgG into these mice. And then not only that, but when these mice were supplemented with N-acetyl D-manazamine, which is a basically sialic acid precursor, the effect was that this increase of the blood pressure was reversed. So they basically again had a blood pressure the same as the control mice, meaning that in this case we actually have this immunoglobulin G, these pro-inflammatory immunoglobulin G glycans, this hyposialylated IgG acting as a functional effector, somehow driving this cardiovascular disease, this hypertension. In humans, we cannot really just reproduce what we saw in mice. It is a little bit more complicated, but it is possible to change a pro-inflammatory profile of glycans in humans. And what we did to basically address this question is we looked at the low-calorie diet and excessive weight loss. And in this case, this was a study where a group of individuals that suffered from obesity was first on a low-calorie diet and then underwent through a bariatric surgery. And the overall effect was that after enough time, they all basically started showing more anti-inflammatory profile of glycans. And this was, the effect was a little bit different between the individuals, with some individuals showing really striking differences, like over 30 30 biological years of difference, but the trend was always the same. The trend was such that with the weight loss, with the bariatric surgery, their IgG profile would get more anti-inflammatory. In another paper that is just accepted, so this is completely fresh, caloric restriction also strongly affected the IgG glycome, and in general, it would generate more anti-inflammatory IgG glycome. However, when these people were put on a weight maintenance diets, which were different diets defined, for instance, as high protein, high GI, high protein, low GI, and so on, there was no general trend. And, the one obvious thing, the one obvious message here is that there is no simple solution. We expected that we would see some general trends in some of these diets, but we did not see them. And most likely there is a strong contribution of individuals' genetics in the effect that we see. One of the things that contributes to, very high interpersonal differences is actually our biological sex. And we can see that women and men actually age differently. So these are pro-inflammatory glycans up here. These are anti-inflammatory glycans. And what we see is that there is around the time of the menopause, when there is a sharp drop in estrogen level. So here in this age group between 40 and 55, that there is a sharp increase in the pro-inflammatory glycans and a sharp drop in the anti-inflammatory glycans in women, while men have a more linear trend of the change. And this was very, very interesting for us, of course. So we went to explore this further. And we saw that basically a chemical suppression of gonadal hormones in a placebo-controlled randomized trial identified a very strong effect of estradiol on this biological age as measured by glycans. And we saw an increase of biological age with hormones, and we saw basically a decrease once their menstrual cycle was recovered. So the changes in IgG glycans during perimenopause normal perimenopause in this case, can be very radical and often precede other symptoms. And we have both scientific data and we have individual examples showing this. So in this case, the measurement was repeated for years, and only around the time of the perimenopause, there was a sharp increase in biological age as measured by IgG glycans. What we think what happens is that IgG glycan integrates estradiol concentration in several weeks, and we're basically seeing an average, sort of a geometrical mean, and we can compare this to HbA1c, for instance, in diabetes, where we're not looking at the glucose concentration, we're looking at the longer time span. And the health effects of the perimenopause are something that needs to be discussed when it comes to the aging, because menopause is often treated as something normal, but we saw that basically there is a huge increase of the chronic inflammation around the time of the menopause and in the perimenopause. And we can see that these effects are actually visible in many, many women, many of them suffering, metabolic syndrome around this time. So we started asking, can there be something done about it? What can we do? And the obvious solution is, of course, HRT. And I'm showing here the preliminary results of an unpublished pilot study where we showed that basically an HRT improves GlycanAge of these people. And, a lot of these lines look very straight, but what I can tell you from these data is also that, a lot of these women that had very good biological age from start, they see more mild improvement. But the ones that had a very bad biological age, they see a drastic improvement. So what we're exploring now is how to use this marker, and can this marker actually be used to individualize HRT therapy. So to conclude, IgG is a biomarker of chronological and biological aging. The inflammaging theory puts IgG glycans at the center stage of healthy aging, and the IgG glycans are functional effectors of inflammation in this case. And IgG glycan can be improved both through lifestyle interventions and therapy, but unfortunately, often there is no one-fits-all solution. Instead, personalized approach is needed, and we believe we have a marker which can allow it. Sex hormones affect the glycans and biological age. Men and women age completely differently. And now we're exploring the option to actually use our biomarker in the hormone optimization and individualized HRT. And with this, I'd like to thank you all for your time. I'm